In this video, we will discuss about how this altered mucin glycosylation helps the cancer cells in its growth and also during metastasis. So we will start with the normal glycosylation of the mucin and the epithelium cells that is shown here. This normal epithelial cells will have normal extended carbohydrate structures on the mucin that can be seen here. However, in the case of cancerous cells, the cancer, because of the uncontrolled growth rate or high growth rate of the cancerous cells, the density of the cancerous cells is quite high, and that will and one more reason is that because of the mutations within the cancerous cells, uh, there is a change in the change in the glycosylation machinery within the cancerous cells, and both of these factor, that is high density and the change in the glycosylation machinery within the cancerous cells, leads to the truncated glycosylation on the mucins that are expressed on the cell surface, that can be seen here. And this some of the truncated glycan structures involve this TN antigen, STN antigens, and this SLEX structure and SLEA structure. And this there two reasons that I have already discussed. Apart from this, the other reasons that might cause this kind of altered glycosylation could be this altered Golgi PS, or this tumor microenvironment might also contribute towards this altered glycosylation because of the high densely crowded area near the cancerous cells. And this altered glycan structure, this altered glycosylation on the cell surface, then helps these proteins, then helps this not proteins, that helps this tumor cells to migrate from uh, one part of the body to another part of the body. And that we can see in the one of the example of this metastasis we can see in the right hand side, in which case the tumor cells that has these altered glycosylation structures on the mucin, truncated glycosylation structures on the mucin, has this SLEX Lewis glycans, and this can interact with the immune cells, and that will trigger this this movement of the beta catenin into the nucleus, and that will lead to the upregulations of this mucin 16 upregulations of the glycosylation of this mucin 16 that is expressed on the cell surface. And this will act as a physical barrier that will prevent these NK cells or natural killer cells to act upon these tumor cells. In other way, this mucin 16 will act as a immune evasion, helps these tumor cells in immune evasion. And this is one of the functions. And other roles that it can do is related to the metastasis, where in the blood vessels this galectin 3 are present, the galectin 3s are present, and it can interact with the T antigen of this tumorous cells, that can be seen here. This galectin 1 right now is not interacted with the tumorous cells, and this tumor cells with the TN antigens are present. After some time, this galectin 3 interacts with the tumor antigen, and galectin 3 has a property that helps in the binding with the endothelial cells. And this kind of tumors that are bind with the galectin then can be interacted with these endothelial cells. And this is one of the reason why cancerous cells are found on the endothelial cells of the blood vessels. And apart from this binding to the endothelial cells, this process also helps these tumor cells to metastasis from one part of the body to the another part, as once it is bind to the endothelial cells of the different organ, it is easy for them to trigger intravasation. And this process in common known as metastasis. And the aberrant glycosylation, that is TN STN structures, on which this galactin 3 binds are usually connected with the usually related to these TNSTN structures on the TNSTN structures on MUC1 MUC1 that is expressed on the tumor cells surface. So this is how the mucin glycosylation helps in the cancer progression, and two of such examples are being explained here. One is related to the immune evasion and another one is related to metastasis. If you need to understand more about this mucin glycosylation role towards cancer progression, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.